Hello, welcome to this session topic from sensor to sensor array, also known as electronic nose. So what is a sensor? Here is a screenshot from Merriam-Webster dictionary that defines a sensor as a device that responds to a physical stimulus, which could be a heat, light, or sound, or any other stimulus, and that stimulus triggers a signal and that signal is transmitted uh, and that's what a sensor means. So again, I'm reading it again: a device that responds to a physical stimulus and transmits a resulting impulse, and that is called sensor. But that definition is too broad, and when we are going to talk about a chemical sensor, so when it comes to a chemical sensor, what the IUPAC, the International Union of Pure and Applied Chemistry, defines the chemical sensor, how it defines. Here is the definition. It is a device that transforms chemical information ranging from the concentration of a specific sample component to total composition analysis into an analytically useful signal. Look at this word, analytically useful signal. Yeah. So any chemical that is present in our sample that should be read out by the sensor, and the readout information should be analytically useful, and that chemical information mentioned above may originate from a chemical reaction of an analyte or from a physical property of the system that is being investigated. So the components of chemical sensors are many, and the two main parts of the chemical sensors are a receptor. The receptor is the one transforms the chemical information into a form of energy, and that can be measured by the transducer. So the second part of the chemical sensor, which is a transducer, is the one that converts the energy from the previous receptor, or from the previous step, into a useful and typically into the form of electrical or analytical signal. A chemical sensor can be classified based on operating principle of the receptor. You remember from the previous slides, like the receptor is the one which initially interacts with the chemicals, or sometimes it is also referred as analytes. Analytes of interest, which means one specific chemical in the mixture of a sample, right? So it can be classified based on the operating principle of a receptor, which is one of that type based on the receptor is physical sensor, in which the signal generated is due to a physical process such as a mass or a change in absorbance or a change in refractive index, or it could be a physical property like a temperature, rise in temperature or a decrease in temperature. Otherwise, it could be a change in conductivity. So when these properties changes, the physical properties of the system changes, and that change is read out by the transducer, then such sensor that were used in to detect chemicals were called physical sensors based on the operating principle of the receptor. A second type of chemical sensor based on the operating principle of a receptor here is chemical sensors. So in contrast to the previous one, where the signal is generated due to a physical process, such as a change in mass or light matter interaction of the system, resulted in the signal. But in this case, a chemical sensor The signal generated is due to a chemical reaction between the analyte molecules and the receptor. So the third classification based on the operating principle of the receptor is biosensors. Biochemical sensors are a subclass of chemical sensors because it is again here the reaction is between the receptor and the analyte is biochemical in nature. So that is why it is a subclass of chemical sensors and the reaction is biochemical in nature, and as a result it could be classified based on the receptor that they were biosensors. Whereas the chemical sensors can also be classified based on the operating principle of a transducer. So if the transducer signaling is based on the optical signal, the optical signal could be of any form or which could be due to several phenomena such as absorbance, fluorescence, luminescence, refractive index. So under this scenario, based on the transduction mechanism, a chemical sensor can be classified as a optical or electrochemical, which could be due to the voltammetric or potentiometric in nature, and otherwise based on the mass it could be a piezoelectric signal, acoustic signals, and the magnetic sense transduction mechanism wherein the paramagnetic properties of the system under investigation is read out and transduced to the output unit as an output signal, or it could be classified as a colorimetric based on the heat effects the system undergoes and which is read out by the transducer. So here we see what are all the factors that could influence the performance of the sensor. There are several factors or parameters that were commonly used in the sensor community, or particularly in the chemical sensor journals or articles or scientific reports. So let's look into those important parameters that will have a either positively or negatively influence the performance of the sensor. First one, sensitivity, selectivity, stability, detection limit, dynamic range, linearity, resolution, response time, recovery time, working temperature, hysteresis, and life cycle. So these are collectively, you could also call them as the key performance indicators or key performance metrics that were used to characterize the uh, sensor or a chemical sensor. So let's see one by one in detail what those key performance indicators are meant to be. You know, sensitivity. It is a change of a measured signal per analyte of interest. Here I have a shortened notion of that is AI, concentration unit, that is the slope of the calibration curve simply. It's quite often confused with the detection limit. So make a note of that. Sensitivity is a change of a measured signal per analyte of interest against the concentration unit. And then the second one, cross sensitivity. So when we talk about sensitivity, it is easier to understand this intuitively that when a sample mixture has more than one analyte and we are interested in one particular analyte among many. So in that case, when we put our sensor, when we expose our sensor to that system, it is going to interact with not just one analyte, but the analyte mixture that is present in the sample. And we are interested in only one out of several. So in that case, our sensor should ideally be responding to our analyte of interest, but not to the others that were present in the mixture. So we expect the resulting signal from other analytes that were not of our interest. Here I call them as interfering analytes, IA. So it doesn't produce any signal, or even if it produces, then the slope of the calibration curve would be much smaller or totally different from the previous one. So in that case, we would be able to discern between our analyte of interest, AI, then compared to the interfering analytes, IA. Hi. So that's what I call it as cross sensitivity. And ideally, we expect that to be a zero for any sensor that we are developing. And what is selectivity? It is a characteristics that determine whether a sensor can respond selectively to a group of analyte, particularly to one analyte, uh, then compared to several other analytes that were present in the sample mixture. Stability. It is the ability of a sensor to provide reproducible results for a certain period of time. So once we have a sensor at hand and which is deployed in for any specific purpose in an industrial application or for environmental monitoring. So how long the sensor could operate as you have expected or tested in the beginning. So that measure or that in the parameter of how long it could provide you reproducible results for the duration of a time, that is called stability. You could say that it is stable for a month or stable for a week or stable for 10 repeated measurements, after which the sensor did not give the same kind of a calibration curve or a line calibration plot. So that's how you say the stability or check or test the stability of a sensor. And it includes retaining the sensitivity, selectivity, and response time and recovery time as well during the period. When we talk about stability, and there are two types of stability, one is called active stability. Active stability is a reproducibility of sensor characteristics during a certain period of time at working conditions, which may even include a high temperature and a presence of a known analyte. So here you will have certain influence from the temperature and during that, under that conditions, you are maintaining the stability for a certain period of time. Whereas the other one is called conservative stability. It is a measure, or it is a property that of a sensor that could retain the sensitivity, the selectivity during a period of time at normal storage conditions such as room temperature and ambient humidity. So you don't have any variations in the temperature, and you still have retained the same sensitivity and selectivity as it was tested in the earlier. Detection limit. It is the lowest concentration of the analyte of interest that can be detected by the sensor under a given set of conditions, particularly room temperature. So what is dynamic range? It is the range of concentration from the lowest detection limit to the highest limiting concentration. Under this range, your calibration curve or the slope of the sensitivity will be remain same, beyond which you might not have a linear relationship. So that range of concentration is called dynamic range, wherein the your sensor would be able to reproducibly give a same result for a signal output for the particular in that concentration range. And linearity is a relative deviation of an experimentally determined calibration graph to an ideal straight line, from an ideal straight line. What's resolution? It is the smallest or lowest concentration difference that can be distinguished by a sensor. You remember in the sensitivity, like when you increase the concentration and you will have a increase in signal as well. So that signal should be increasing signal should be statistically valid and significant for the uh, change in the concentration. That difference, that smallest concentration difference that could yield a statistically significant signal is called resolution in sensor. Response time is a time required for a sensor to respond to a step concentration. I mean, from one concentration to the other concentration, if you increase, how much time it takes to give the statistically significant signal, and that time is called response time. Recovery time is the time it takes for the sensor signal to return to its initial value after the step concentration uh, um, from a certain value to zero. For example, for each aliquot of analyte introduction, you will expect to change or see an increase in the output signal. So after adding the second or third aliquot, the time it takes to come back to the normal, the baseline, is called recovery time. And what is a working temperature? It is usually the temperature at which uh, the maximum sensitivity is obtained for the sensor. Hysteresis, that is the maximum difference in output when the value is approached with an increasing and a decreasing analyte concentration. What is life cycle? So it is the total life or a period of time for which the sensor would be able to operate continuously. So here, what kind of values would you like to have in ideal chemical sensors? For example, the key performance matrix sensitivity, we would like to have a higher value so that for a smaller change in concentration, you expect a very high or the difference in the signal is higher. And the selectivity, you want that to be very high, which means you don't want the cross sensitivity is zero when you have a high selectivity, or other words, your sensor is specifically responding to one particular analyte in the presence of interfering analytes or in presence of a mixture of analytes that were present in the sample that is being tested. Dynamic range, you would like to have a very high dynamic range so that you could detect from the lowest detection limit to the higher concentration that were either in requirement for the sensor operation or where the sensor is going to be or will be deployed. Stability, you want that to be high, and the life cycle, you want that to be a longer or a longer period. So, um, an ideal chemical sensor, what are all the desired values for a response time? So you would like to have a very small value for the response time, and a low or a smaller value for the detection limit, and a smaller value for the hysteresis, and a smaller deviation from the linear. But when we are talking about ideal chemical sensors, would it be possible or practical to have or meet all those key performance metrics to those desired value in a practical sensor? No, not necessarily. I mean, in a practical sensor, you don't need to meet all the requirements of an ideal sensor, but instead focus on the requirements. For example, here we are thinking about a sensor that is going to be deployed in an industrial application to control a process or to monitor a process wherein the chemicals that were present in the reactor would be much higher than the sub parts per billion or sub ppb parts per million ppm. So you don't need a very small or a low detection limit in that case when you already have a molar or millimolar concentration range that is present in the reactor. So you don't need to worry about the lower detection limit in that case. But it would be indeed favorable to have a small or a very small response time than compared to a lower detection limit. So that is how you kind of balance uh, you know the requirements or trade off few things against what is in fact or indeed is essential for the successful operation or demanded requirements for the particular sensor. Here in this case, we are considering about a sensor that is needed for environmental monitoring. You know, so in that case, your response time might not be a stringent criteria, whereas you would like to have a very low detection limit because in the environmental application or in environmental monitoring, you will be having a open space or probably a constrained or a semi-constrained space in which the emitted gases or it could be in a liquid form. So the emitted analyte would be very small compared to the existing sample volume or yeah. So for that reason, you would really like to have a very low detection limit for the sensor that you're developing for the environmental application. And with this, I would really like to thank you all for your attention, and we will see again with more examples of each, or at least for the chemical sensors, mainly focusing on the electrochemical sensors in the following uh, time or in the following period. Until then, thank you. Bye.